Hello everyone, I am Zoeb Mohammad Zia, Assistant Professor at the PG and Research Department of Zoology, the New College, Chennai. Today this video is going to be a short overview, sort of a snapshot overview of this topic, which is somatic hypermutation and class switching to generate antibody diversity, or the Vdj recombination and the isotype switching happening in the B lymphocyte cells to generate antibody diversity. Now, before looking at the generation of the antibody diversity, we look at the generation of the B lymphocytes. As you all must be knowing, the B lymphocytes are produced and also matured inside the bone marrow. So they are produced from the hematopoietic stem cells. They go through all these stages and finally they mature in the bone marrow. Once they are mature, they circulate through the bloodstream into these peripheral lymphoid organs or the secondary lymphoid organs like the spleen, where they are activated by antigen stimulation. And once they are activated, they differentiate into these immunoglobulin secreting plasma cells and also the memory cells. And once they differentiate into plasma cells, they can also undergo class switching to produce any of the 5 classes, that is the 5 isotypes of the antibodies. So this diagram here is a very familiar diagram showing the structure of a immunoglobulin, that is the antibody. So here you can see it has 2 chains. This one in orange is called the heavy chain and this one in blue is called the light chain. Both these heavy chains as well as the light chain will be having a variable region represented here as V and a constant region shown here as C. So CH is a constant region of the heavy chain and Cl is the constant region of the light chain. Similarly, VH is a variable region of the heavy chain and VL is a variable region of the light chain. So here maximum recombination, maximum variation, what you see is in this variable region. The constant region will remain almost constant, that is no recombination, no variation. Variation will be seen in the variable regions, which are also the antigen binding locations. So this is where the antigen will be binding and these will be highly variable, whereas the rest of it will be almost constant. And here again I have mentioned the isotypes. You have 5 isotypes of 5 classes, that is IgM, IgD, G, E and A. All of these classes are actually caused by the constant region of this heavy chain. For example, if this constant region of the heavy chain consists of a new chain, it will be a IgM. If it had a delta chain, it will be IgD. If it had a gamma chain, it will be IgG. Epsilon chain, it will be IgE, and alpha chain, it will be IgA. Now to look at the recombination, that is a variation. I told you the variation, the recombination maximum, it will be in this variable region. The constant region will be the same. Now we look at the recombination first in the light chain. So out of these two chains, the heavy chain and the light chain, the first recombination we are going to see is in this chain, the light chain. So the light chain recombination is going to be called the Vdj recombination. That is, it has a variable segment V and the joining segment J. Now as you can see in this diagram, this variable segment is going to join with the joining segment. So this can be more than 30 of these segments can be there, and more than 4 of these segments can be there. So you can join any of this V segment with any of this J segment, and always remember this V segment will have a leader segment at the 5' end. So this black segment what you see given as L is a leader segment, and I'll tell you what is the importance of that leader segment. So here the first step is recombination between V and J. So V and J join along with the leader, and then the next step is this Vj recombines with C. You have only one C, so there is no variation in that. So whatever variation is there, it will be there only in the Vj. No variation in the C because C is only one. So this Vj combines with C. Remember there was a leader segment along with the V segment. Here what happens after the translation of the nascent polypeptide, that is the polypeptide is complete, but it's not a protein yet, it is only a nascent chain. So what happens here, this leader, it pulls out the protein from the endoplasmic reticulum. As you must be knowing, obviously the protein is produced inside the endoplasmic reticulum. Now from the endoplasmic reticulum, this leader, what it does, or the uh, signaling peptide, what it does, it just pulls out the peptide fragment. The peptide fragment it pulls out and it is cleaved. So now this peptide sequence is produced combining of the V, that is a variable segment, and the joining segment and the constant segment. V and J together give rise to variable region of the light chain. This is a kappa light chain, and C gives rise to constant region of the kappa light chain. You must be knowing there are two light chains. One is a kappa light chain and one is a lambda light chain. This example what we saw here was the kappa light chain. So here it begins in the germline DNA of the kappa light chain. V and J combine. Next step, Vj combines with C, and finally you get the variable kappa region and the constant kappa region. So here you have, in this region you have V and J segments combined to give this region. This region in light blue is made of V and J segments combination, and this you see in dark blue is made of the constant region. So that was a recombination in the light chain. Now we see the recombination in the heavy chain. So almost similar, but a little more complex than the Vdj recombination. It is called Vdj recombination. So this is the variable segment, diversity segment, and the joining segment. So these three segments come together to form the variation in the heavy chains. So again, as we see, that is going to be a leader segment preceding the variable segment, and you have the joining segment, and the only difference here from the light chain is we have intervening segments of the diversity kind. So D is intervening between V and J. Rest of it is almost same. So here the first step is recombination or joining between D and J. Again, you have many D segments, that is diversity segments, and many J, that is joining segments. So through permutations and combinations, uh, any one of this D segment can recombine with any one of these J segments, that is a joining segment, to give a DJ segment. And next, any one of this variable segment can combine with a DJ segment. So any one of this, but along with the leader segment. Always remember the variable will be preceded by a leader segment. So now this Vdj recombine, and this Vdj is going to recombine with both of these constant segments, constant segment nu as well as constant segment delta. So it's going to express both uh, immunoglobulins, both antibodies. What are they? Nu is IgM and D is IgD. So you can see in this diagram here at this stage, when it first matures, it's going to express both IgM as well as IgD. IgM is denoted by the constant region mu, that is C mu, and this will be constant region delta, that is C delta. So in this diagram you see both constant regions. First step, you've got the Vdj recombination, then it's going to recombine with both of these nu and delta, and once it is activated by a type of RNA splicing, it's going to express any one. So if it expresses nu, you get a new heavy chain which gives IgM. If it expresses constant region delta, it will express the delta heavy chain which gives IgD. So here in this diagram we saw this part, this light blue is going to be produced by recombination of what segment? V and J. Vj segment is going to give the variable region of the light chain, whereas the Vdj segments recombination is going to give the variable region of the heavy chain. The constant segment of the light chain will give this constant region, whereas the constant segment of the heavy chain is going to give the constant region of the heavy chain. And finally, these are the number of segments. These light chains, as I mentioned, they are of two types, the kappa type and the lambda type, and both are on different chromosomes. So if it is a kappa light chain, there can be about 34 to 38 variable segments, V segments it can be. 34 to 38. So it can take any one of this V segment and recombine with J segment. I told you light chain will have Vj recombination. Heavy chain will have Vdj recombination. So first for the Vj recombination, you take any one of these V segment and you recombine with any one of these J segments. So you have 5 choices. You take any one of these and recombine with any one of this, and finally recombine with the C. I told you there's no variation or very little variation in the constant segment, that is the C segment. And similarly, you make a heavy chain. Take a V, you take a D, and you take a J, and you recombine these, and along with that you take a C. C is a constant, so very little or no change in the constant region. And through this, by this selection process, you are able to generate the antibody diversity, all different type of antibodies. And now for the class switching, there is a mechanism where there are regions in between these C segments, these constant segments. You see in between them there are segments which will have switch regions or switch segments. What these switch segments do is, if there is a switch segment here and a switch segment here, what happens once it is activated, the B cell, once a B cell is activated by a antigen, and you know what class or what isotype is to be produced, this switch region and this switch region, they just loop together, and what happens, this switch region goes into this place, and here you get a new switch region which recombines with the Vdj. So you will be getting a different class of molecule, and ultimately what happens is you get different affinities of antigens on the same isotype. For example, IgM, it might have a different uh, variable region. The another IgM might have a different variable IgM. So the same class of antibodies can have different affinities due to class switching. Now this whole video what you have seen is very preliminary topological understanding of this, and it is not understanding of the process or the mechanism that happens during recombination. This is just for your understanding.